Close your eyes for a moment and hum. A single sustained note, at whatever pitch feels natural, with your lips gently closed, for about 10 seconds. Notice what happens. A vibration spreads through your skull. Your chest resonates. There is a quality to the sensation that is difficult to name precisely, not quite calm, not quite alertness, but something occupying the space between the two. You almost certainly do not know the mechanism behind any of what you just felt. Almost nobody does, because the neuroscience and physiology of humming has never been explained in popular terms despite the fact that the research behind it is genuine, replicated, and specific enough to tell you exactly what was happening in your sinuses, your vagus nerve, your brainstem, and your prefrontal cortex during those 10 seconds. This video traces that mechanism completely, from the physical acoustics of the sound itself through the neurological pathways it activates. And by the end you will have a precise, mechanistically grounded understanding of why humming produces the specific sensations and physiological changes it does, and why the response is considerably more interesting than a simple relaxation effect. To understand what humming does to the body, you first need to understand what humming physically is, in acoustic and mechanical terms, because what makes it biologically distinct from other vocalizations is not its emotional content, or its pitch range, but a specific physical property of the sound it produces. It is voiced, resonant vibration transmitted simultaneously through air and through solid biological tissue. When you hum with your lips closed, your vocal folds vibrate, producing sound waves in the range of roughly 80 to 500 Hz depending on your natural pitch, which would normally travel outward through an open mouth. With the lips closed, the primary outward escape route for that acoustic energy is blocked, and the sound is redirected through the nasal cavity and through the bones and soft tissues of the skull, which resonate at the frequencies being generated and transmit the vibration as both airborne sound through the nasal passages and as bone-conducted vibration propagating through the craniofacial skeleton. This combination of resonance pathways is what produces the felt sense of the sound being inside your head when you hum, and it is also what places the vibrating air column in direct contact with specific anatomical structures that are not reached by normal externally directed vocalization. The paranasal sinuses, the air-filled cavities in the bones surrounding the nasal passages, including the maxillary sinuses in the cheekbones, the frontal sinuses above the eyes, and the ethmoid and sphenoid sinuses deeper in the skull, are all connected to the nasal cavity through small openings called ostia, and the air columns within them are set into oscillation by the pressure waves generated during humming, in a physical phenomenon called acoustic coupling. This sinus resonance is not incidental. The air within those sinus cavities has a biochemical composition dramatically different from ambient air during humming, and what happens to it when it is acoustically agitated is the first major mechanistic story of this exploration. Before that sinus story, however, there is a neural pathway activated by humming that is even less widely known and even more precisely documented, the stimulation of the vagus nerve through its auricular branch, the only peripheral branch of the vagus nerve that runs to the surface of the body outside the torso. The auricular branch of the vagus nerve, sometimes called the Arnold nerve, supplies sensory innervation to the floor of the external auditory canal, a small area of the skin inside the ear, and to a portion of the outer ear. This branch is genuine vagal tissue, carrying sensory fibers whose cell bodies sit in the vagal ganglia, and whose central projections terminate in the nucleus tractus solitarius in the brainstem, the same nucleus that receives all other vagal sensory input from the heart, lungs, and gastrointestinal tract and that coordinates the parasympathetic homeostatic responses associated with vagal activity. Auricular vagus nerve stimulation, the deliberate activation of this ear branch to produce centrally mediated vagal effects, has been studied in clinical research as a non-invasive alternative to implanted vagal nerve stimulators, primarily because it offers access to genuine vagal afferent fibers without surgical intervention. Studies examining auricular VNS using small electrodes placed in the ear canal have demonstrated effects including heart rate reduction, heart rate variability increase, and changes in brainstem and cortical activity consistent with increased parasympathetic tone. And this is where the humming connection becomes mechanistically interesting, the bone-conducted vibration produced during humming propagates through the craniofacial skeleton and through the cartilage and bone surrounding the external ear canal, producing mechanical vibration of the ear canal walls and the skin lining them, including the small area of skin innervated by the auricular branch of the vagus nerve. Whether this mechanical vibration during humming produces auricular vagal stimulation of sufficient magnitude to generate measurable autonomic effects is a hypothesis with growing mechanistic support and with indirect empirical backing from the heart rate variability research on humming we will examine in detail shortly, but it has not been tested with the same directness as electrical auricular VNS in controlled research settings. It represents a plausible, anatomically grounded mechanism that sits alongside the more directly documented effects of humming on the respiratory pattern, which also engages vagal pathways through a separate mechanism. Humming produces a sustained, controlled expiratory phase that extends the breath significantly beyond its resting duration, and extended exhalation, as explored in the deep breathing discussion, drives vagal activation through the respiratory sinus arrhythmia mechanism. Regardless of the acoustic component of the vocalization, these two pathways, auricular vibration and extended expiratory phase, may each contribute to the autonomic effects of humming through partially independent channels simultaneously. The sinus nitric oxide story is the most specifically documented and most surprising finding in the physiology of humming, and it begins with a measurement almost nobody outside of phoniatric and otolaryngological research ever encounters. In a study published in the American Journal of Respiratory and Critical Care Medicine by John Lundberg and Eddie Weitzberg, researchers measured the concentration of nitric oxide in the exhaled air from the nasal cavity under two conditions: normal nasal breathing with the mouth closed and humming with the mouth closed. The difference was not subtle. During humming, nasal nitric oxide concentration increased approximately 15-fold compared to quiet nasal breathing in the same subjects, with the oscillating airflow produced by the humming vibration dramatically accelerating the exchange of gas between the paranasal sinuses and the nasal cavity through the sinus ostia. The mechanism driving this 15-fold increase is straightforward once the anatomy is understood. The paranasal sinuses are potent producers of nitric oxide through the enzymatic activity of nitric oxide synthase in their mucosal lining, and they accumulate high concentrations of nitric oxide in their essentially stagnant air volume during periods when the sinus ostia are functioning, but airflow through them is minimal. During normal quiet nasal breathing, gas exchange between the sinuses and the nasal cavity occurs relatively slowly through diffusion and the minor pressure fluctuations of breathing. During humming, the oscillating pressure waves generated by the vibrating vocal fold column create repeated, rhythmic pressure fluctuations in the nasal cavity that pump air in and out of the sinus ostia in a much more vigorous pattern, rapidly flushing the accumulated high-concentration nitric oxide out of the sinus cavities and into the nasal airstream, where it is inhaled into the nasal passages and the upper airways. The significance of this nitric oxide flush extends beyond simple measurement. Nitric oxide in the respiratory tract serves multiple functions including bronchodilation, the relaxation of smooth muscle in airway walls that reduces resistance to airflow, and antimicrobial defense, since nitric oxide is directly toxic to certain bacteria and viruses at the concentrations found in the sinuses. The delivery of sinus-derived nitric oxide to the nasal and upper respiratory passages during humming may therefore have local immune and airway function effects beyond the systemic cardiovascular effects of nitric oxide entering the bloodstream. Though the research specifically documenting clinical outcomes from humming-derived sinus NO delivery in healthy subjects remains limited compared to the underlying measurement of the NO concentration change itself. What is better documented is the passage of at least a fraction of this nasally inhaled nitric oxide across the olfactory epithelium and potentially into the olfactory nerve pathways, which have anatomical access to specific brain regions not easily reached by systemically circulating nitric oxide due to the blood-brain barrier. Nitric oxide is a highly lipid-soluble, membrane-permeable gaseous signaling molecule that can diffuse freely through cell membranes wherever it encounters them, and its presence in high concentration in the nasal cavity during humming places it in proximity to the olfactory epithelium, whose nerve fibers project directly into the olfactory bulb and from there into the piriform cortex, the amygdala, and the hippocampus, a direct neural chemical route to limbic brain structures bypassing the vascular route through which most neuroactive substances are delivered. Whether nasal nitric oxide during humming actually reaches these brain structures in biologically significant concentrations is a mechanistic hypothesis that the current research does not yet fully confirm, but the anatomical plausibility is direct and the measurement of the nitric oxide concentration increase driving it is solid. The changes in brain activity during sustained humming documented in neuroimaging research represent the most direct window into what the brain is actually doing during this vocalization, and what functional MI studies examining vocalization and chanting have consistently found is a pattern of activity that differs in specific, interpretable ways from both silence and from spoken language. Sustained humming and similar tonal vocalizations produce measurable reactivation of the default mode network, the collection of brain regions including the medial prefrontal cortex, the posterior cingulate cortex, and the angular gyrus that is active during self-referential thought, autobiographical memory, and the mind-wandering state characteristic of unfocused rest. This reactivation is paradoxical at first glance because the default mode network is typically suppressed during externally directed task performance, and humming is a deliberate voluntary action. The resolution of this paradox lies in the specific attentional character of humming. It occupies motor and auditory processing systems with a repetitive, internally generated stimulus that is sufficiently automatic to require minimal executive control, leaving the default mode network free to engage in its characteristic self-referential processing while the vocalization continues as a kind of anchor. At the same time, sustained vocalization is associated with increased coupling between the prefrontal cortex and the amygdala, the connectivity between these regions being a marker of top-down emotional regulation. The prefrontal cortex's capacity to modulate the amplitude of amygdala threat responses. This increased prefrontal-limbic coupling during sustained vocalization has been proposed as a mechanism contributing to the mood and anxiety effects of singing, chanting, and humming practices documented across clinical research, since the amygdala's response to perceived stresses is attenuated when its activity is under stronger prefrontal modulatory influence. The proprioceptive dimension of humming, the felt sense of vibration inside the skull, the resonance in the chest, the sensation of the sound being generated from within rather than arriving from outside, deserves its own mechanistic attention because it is a genuine sensory experience with documented neural correlates rather than a subjective impression without biological substance. Your larynx, the structure housing the vocal folds that produce the sound during humming, is densely innervated with mechanoreceptors embedded in its mucosa, muscles, and ligaments that detect the specific mechanical deformations produced by vocal fold vibration during vocalization and transmit this information through the superior laryngeal nerve and the recurrent laryngeal nerve to the nucleus tractus solitariae and the sensory thalamus, where it contributes to the brain's proprioceptive map of the vocal apparatus. This laryngeal proprioceptive feedback is not merely technical monitoring of the acoustic output. Research on the neural correlates of singing and sustained vocalization has found activation of the anterior insula, a region associated with interoceptive awareness, the sensing of the body's internal state, during sustained tonal vocalization, consistent with the interpretation that the proprioceptive feedback from laryngeal mechanoreceptors contributes to the felt quality of humming as a whole-body internal experience rather than purely an auditory one. The bone-conducted vibration that spreads from the larynx through the cartilages of the trachea, the sternum, the ribcage, and the craniofacial skeleton during humming activates mechanoreceptors distributed throughout these tissues simultaneously, creating a distributed proprioceptive experience that is qualitatively unlike the experience of any externally generated sound of equivalent loudness, because the vibration is being sensed from within the vibrating structures themselves rather than arriving at the cochlea as airborne pressure waves from an external source. The heart rate variability research on humming provides the most direct physiological measurement available for quantifying its autonomic effects in aggregate form. Studies measuring heart rate variability, the beat-to-beat variation in heart timing that reflects the balance of autonomic input to the sinoatrial node during humming and comparative control conditions have found consistent increases in high-frequency heart rate variability, the frequency band directly associated with respiratory sinus arrhythmia and therefore with parasympathetic vagal cardiac influence, during sustained humming relative to normal quiet breathing at the same respiratory rate. This finding is consistent with the auricular vagal stimulation hypothesis, with the extended expiratory phase mechanism, and with any additional autonomic effect of the nitric oxide release or neural resonance pathways operating simultaneously. And it provides an outcome-level measurement that these mechanistic hypotheses collectively predict. Research on the brainwave changes accompanying sustained humming and similar tonal practices has used electroencephalography to document increases in alpha wave power, the 8 to 12 Hz oscillation associated with relaxed, unfocused alertness, and with reduced cortical arousal during humming compared to rest in multiple studies, with some research also finding increases in theta power, the 4 to 8 Hz range associated with meditative states, memory encoding, and the hypnagogic threshold between waking and sleeping. These brainwave changes are consistent with the fMRI findings of default mode network engagement and prefrontal-limbic coupling, together painting a picture of a brain state during sustained humming that is simultaneously internally focused, emotionally regulated, proprioceptively rich, and less aroused in its cortical activity than normal baseline waking, a combination that subjectively maps onto exactly the quality of experience most people report during sustained humming, a state that is calm without being drowsy, inward without being dissociated, and physically satisfying in a way that is difficult to attribute to any single sensory channel. The universality of humming across human cultures, its spontaneous emergence in infants before language acquisition, and its presence in the ritual and contemplative practices of societies with no historical contact with one another, from the OM of Indian meditative tradition to the throat singing of Tuvan practitioners to the wordless humming that emerges spontaneously in humans engaged in comfortable, repetitive work, suggests that this is not a culturally transmitted behavior but a biological one, an instinctive exploitation of the physiological responses we have just traced, driven by the reinforcing sensations those responses produce. The beta-endorphin production discussed in the sunlight context finds a parallel here in the opioid-adjacent sense of reward that accompanies pleasurable vocalization, and the vagal activation and HRV increase find a parallel in the calming effect that makes humming one of the most universally practiced self-soothing behaviors across the human lifespan. From the mother instinctively humming to a distressed infant to the adult humming while preparing a meal at the end of a long day. If you want a protocol grounded in everything this mechanistic exploration has established, here it is, and it is remarkably simple. 60 seconds of sustained humming at a comfortable pitch, through the nasal cavity with the mouth closed, produces the following in documented sequence: the acoustic agitation of the paranasal sinus air columns, initiating the nitric oxide flushing mechanism that Lundberg and Weitzberg measured at 15-fold baseline concentration in the nasal air, mechanical vibration of the external ear canal walls, stimulating the auricular branch of the vagus nerve and contributing to the parasympathetic shift measurable as increased high-frequency heart rate variability, an extended expiratory phase relative to resting breathing, activating the respiratory sinus arrhythmia vagal mechanism independently, distributed proprioceptive activation of laryngeal, tracheal, and craniofacial mechanoreceptors contributing to the insular-mediated interoceptive richness of the experience, and a brainwave shift toward alpha and potentially theta power consistent with a relaxed, internally focused, emotionally regulated neural state. All of this in 60 seconds. None of it requiring any equipment, any training, any cost, or any prior knowledge of the mechanisms producing it. So here is the real reframe to sit with as we close this exploration. Every time you have ever hummed absent-mindedly, while working, while cooking, while sitting in traffic, while holding a child, you were not simply making a sound. You were activating a neurophysiological circuit that your biology built into the human vocal apparatus not as a byproduct of language, but as an independent, ancient mechanism for vagal stimulation, sinus ventilation, proprioceptive interoception, and autonomic self-regulation. The calming effect you felt was not imagined. The resonance in your skull was not merely acoustic. The mechanism running beneath both of them is specific, it is documented, and it has been waiting inside your own larynx your entire life for someone to explain it precisely. Hum for 60 seconds right now, before the next video starts, and notice the mechanism as it happens. Leave us a comment telling us what sensation during humming surprised you most once you understood what was producing it, and subscribe so you never miss the next time we look closely at the extraordinary physiology hiding in the ordinary things you do every day. Until then, keep questioning the things you assumed you already understood, because the most remarkable biology in your own life is very often the biology making the quietest sounds.